Meiosis is also called a reduction division because chromosome numbers become 1/2 in each daughter cell. Meiosis consists of two stages, meiosis I and meiosis II. In meiosis I, actually, meiosis I is technically the reduction division because during meiosis I, the chromosome number becomes 1/2 and two daughter cells are formed from one mother cell. The meiosis II occur in both of these daughter cells separately, and both of these daughter cells, each one divides into two further. So ultimately, at the end of meiosis, we have 4 daughter cells. But the division, the actual reduction division in which chromosome number reduced to 1/2 is the meiosis I. Meiosis II is just like that of mitosis. Now we talk about meiosis I. Meiosis I consists of a prophase. We call it prophase I because this is the prophase of the meiosis I, metaphase I, anaphase I, telophase I, and a cytokinesis. When the cell divides into two, then when the nuclear division is complete, then the cell divides into two by cytokinesis, and when it is almost divided, it starts its next phase of division called meiosis II. Meiosis II is just like the mitosis. It simply divides both the cells into into same type of cells with equal number of chromosomes. Now we look at the detailed process of meiosis. Meiosis I. Meiosis I consists of prophase, then a metaphase, then anaphase, then a telophase. We look at first of all the prophase I. As we know that the chromosomes, they are just like mitosis, they are already duplicated. It means that every chromosome have two sister chromatids. It is doubled in the number. As you can see in the diagram, there are two homologous chromosomes which are shown. Each chromosome, which is now called each homologue will consist of actually two sister chromatids. You can see one in green and the other in blue. The green one is one homologue, that is one chromosome which is duplicated. The blue one is its homologous partner, and which is in its appearance just like that of the first one, which is shown in blue color, and this also consists of two sister chromatids. Now what happened in the prophase I, as is shown in the second diagram? The prophase I is the longest phase of meiosis. In this phase, actually, the homologous chromosomes, they pair up with each other. They come close together, and some of their parts attach to each other to make the structures called chiasmata. In the diagram, the second diagram, you can see a single chiasma in which one part of homologue 1 is attached with the, or we can say crossed with the part of the homologue 2. We call this process pairing up or synapsis. We say that these chromosomes, the homologues, they are synapsed together. But remember at this stage that only the homologous chromosomes will pair up with each other. As we know that in human beings, for example, they have 46 chromosomes, and there will be 23 pairs. The chromosomes in one pair only will make chiasmata with each other. One homologous chromosome will not pair up with any other homologous set. They will only pair up with their own type. If they do so wrongly, that will be an error in the meiosis, which may result in a problem later on. So usually or normally, homologous chromosomes pair up with each other. We call them these pairing synapses. Now what is the purpose of this pairing? Purpose of this pairing is recombination. These parts of chromosomes which pair up, which are crossing each other, what will happen that these parts, both of these parts, will break up from their original chromosome and will attach to the other homologue. As you can see in the bottom of the diagram, now the two homologues are shown, but you can see that the homologue on the top in the green color have a small part of blue color, which is actually broken up during the chiasmata, during the synapses from the homologue 2 and attached to the homologue 1. Same happened with the homologue 2. You can see in the last part of the diagram that the one part of homologue 2 looks like green, because actually this is the part of homologue 1 which was during chiasma again, the pairing up, broken up and attaches to the homologue 2. Now you see that both of the chromosomes, they have a large part of each other. They exchanged their parts. We say that they exchanged their DNA or nuclear material. The result of this situation is new recombinations. There are new sets of genes which are formed, and the result will be new characteristics which will appear in the organism. This is the most important property and advantage of the sexual reproduction, that during the meiosis process, the cells, the chromosomes, they do recombine. And as we know, as many chromosomes as an organism have, as many recombinations it can form. And every recombination may result into a different characteristic, which gives an evolutionary advantage to that particular organism. Meiosis, the prophase 1 is one of the most important parts of meiosis. These paired chromosomes which are making pair, as you can see in the diagram on the top, these chromosomes, homologues which are paired, the homologue 1 and homologue 2, collectively are called sometimes bivalents or tetrads. They are called bivalents because both homologues are attached with each other. They are called tetrads because we know that due to duplication or the replication, each homologue consists of now 2 sister chromatids. So in total, they are making 4 chromosomes. We call them tetrads, or sometimes we call them bivalents. Till end of the prophase 1, lot many recombinations like this are formed, constituted, because almost every homologous pair up with each other, and they exchange their different parts. So lot many recombinations they are formed. With this, all the other features of the prophase, they also takes place. The spindle fibers are formed because chromosomes have to attach to spindle fibers to move from their place, from the center towards the poles. So spindle fibers are formed. In animal cells, centrioles will do it. In plant cells, spindle fibers will be formed by other methods. And the nuclear membrane, ultimately at almost end of this phase, will disappear. Now there is one more important difference. Now each homologous pair is attached with each other by one kinetochore. There is one kinetochore per each homologous pair, which is in contrast to mitosis, in which there is one kinetochore per one sister chromatid. Now both sister chromatids of one homologue are attached with only one kinetochore. And this is very important because we know that meiosis is the reduction division, and one homologue have to go to one pole and the other homologue have to go to the other pole. Now we know that at about end of the prophase, spindle fibers which are already formed, each homologue attaches to the spindle fibers with its kinetochore. Now, because there is one kinetochore per homologue, so every homologue as a whole is attached to a spindle fiber. Which is same is true for the other homologue. There is one more important thing. As we know that every organism which is sexually reproduced always have one set of chromosomes coming from the mother and the other set coming from the father. The homologues which are coming from mother and father, they are arranged in different orientations. That is, in one pair, for example, the maternal homologue will be on one side towards one pole, and the paternal homologue will be on the other side. This is different for each chromosome. It means that in a gamete, the there are about 50-50 chances that the gamete will have a maternal homologue or a paternal homologue, which is also a very important feature for we can say new recombinations. And at the end of when the spindle fibers are formed, the chromosomes they attach, the nuclear membrane is dissolved or start disappearing, and it is disappeared at the end of very long phase, the prophase 1, and then the prophase 1 ends. When the prophase 1 ends, the next phase starts, that is metaphase 1, because this is part of the meiosis 1. Now in the metaphase 1, the chromosomes, the homologues, they are attached on the spindle fibers, arrange themselves on the equator. All of the homologues are arranged on the equators almost in a rough straight line or in rough circular we can say part. After the metaphase comes metaphase 1, comes the anaphase 1. Now anaphase 1 is very important, because in this anaphase 1 of the meiosis, the chromosomes have to move towards the pole, and now half chromosomes have to go to one pole and the half chromosomes have to go to the other pole. What happened that because we know that there was one kinetochore per homologue, per two sister chromatids. It means that the two sister chromatids are going to move towards one pole, and the other two sister chromatids are going towards the second pole. Now the microtubules or the spindle fibers, they start pulling the chromosomes towards the poles of the cell, now because again I repeat the same thing, because there is one kinetochore per two sister chromatids of one homologue. So one homologue, which consists of now two sister chromatids, will move towards one pole, and the other homologue, we can call here homologue 2, which also consists of two sister chromatids, will move towards the other pole. So the half set of the chromosomes is moving towards one pole, and the other half set is moving towards the second pole. So this is the anaphase 1. Now this stage is extremely important, because if there is a slight error in this phase, it results into an unequal distribution of the chromosomes in the gametes. And if there is an unequal distribution of chromosomes in the gamete, it results in very serious disorders. When the anaphase finishes, the chromosomes are moving towards poles, almost they are reaching near poles. The last stage of meiosis 1 starts, that is called the telophase 1. In telophase 1, the half number of chromosomes reached at one pole, the other half of chromosomes half set is reached at the second pole, and now the nuclear membrane starts forming, and the chromatin material start slightly decondensing. With this, the meiosis 1 or the nuclear division 1 of meiosis is complete, and then the cell is divided into two cells, in again just like in plant cells by a phragmoplast, and in animal cells by a cleavage furrow formation. Cell is divided into two. Now there is a slight difference from mitosis, that what happened that sometimes these cells are completely divided into two. When they are just dividing at the end of their cytokinesis, they just start their second nuclear division, which is called meiosis 2. Because meiosis is a continuous process. So what happened that when the nuclear division is complete, cleavage furrow is formed, and cleavage furrow almost reaches at the end, then the next nuclear division just starts. We call it meiosis 2. Now we have a look on a diagram which explains the meiosis 1. If we look at the diagram, the first part shows the interphase 1. When the cell is at interphase, its chromosomes, a chromatin material, is not very visible, and it is showing a centrosome, a pair of centrioles. The next diagram towards right, you can see that the chromosomes, the homologous pairs, they consist of 2 sister chromatids each, and they are forming chiasmata. Now here in this diagram you can observe that per homologue more than one chiasmata could be formed. You can see in the small sized chromosome pair, the homologue pair, there is one chiasmata, but you can see the chromosome which is larger one, there are 2 chiasmata. So it means there may be more than one chiasmata per homologous pair. Then comes the next stage, the metaphase 1. In the metaphase 1, as we talked about it, in it the chromosomes are arranged on the equatorial plate in the center, but now each homologue is attached with the kinetochore to the microtubules, the spindle fibers, and not sister chromatids. In anaphase 1, you can see that the whole homologue, the complete chromosomes with its both sister chromatids, is moving towards the poles. And you can observe various recombinations present in these chromosomes which are going towards the poles. Then comes the telophase, they start decondensing, nuclear membrane starts appearing, and the cell divides.